Vibrio cholerae typically involves isolating the microbe from a fecal sample on thiosulfate citrate bile sucrose agar, or TCBS. It's probably not the best medium out there because there are some other chromogenic media. I think Hardy Diagnostics might have one as well that might be superior to TCBS, but it works reasonably well. And so the mode of action is basically it's a selective and differential medium to distinguish cholera from other types of bacteria that could be in a water sample or a fecal sample primarily. So the medium contains thiosulfate and citrate and has a really high pH. And so the Vibrios like this don't mind that high pH. But the Enterobacteriaceae, so microbes like E. coli, which would be abundant in fecal samples, do not like that, nor do they like thiosulfate or citrate. There's also bile and cholate, bile salts, to suppress enterococci. So enterococci, the so-called fecal strep, that could contaminate a plate and overrun your desired target microbes. The medium also contains sucrose and a pH indicator, thymol blue and bromothymol blue, that are used to detect the acid byproduct of sucrose fermentation from Vibrio cholerae cells. And so the medium actually looks kind of green like this. And then when you do a nice streak plate of the organism onto this medium, you get this yellow halo representing the sucrose fermentation. So the fact that the microbes grew suggests that they're cholera, but also the fact that there's a yellow halo would also suggest. And so you can combine this information here with the clinical manifestations of disease. So a watery diarrhea that you can then dip into and do platings onto TCBS. And if you get these data here, you can have a positive ID. Another way to ID these organisms and also to serotype them is through immunohistochemical staining. So in this particular case, this is just a cartoon representation of the cell surface of the bacterium. And so you can see different antigens on the surface of the bacteria. And so in our case, the antigens would be those sugars sticking out from the O antigen in the LPS of the outer membrane. And so you would have different classes of antibodies labeled with a fluorescent fluorophore, and the antibody can then be applied to the microbe, just like doing a simple stain, for example. The microbe is applied to a slide as a smear, and then you can flood the slide with these fluorescent tagged antibodies. And if you get binding, as you see here, and then under an epifluorescence microscope, just turn the lights off and flip on the right wavelength of UV, the cells should glow. And if there's no reaction to that particular antibody, this whole field would be completely dark. So this is one that a student of mine did, just so we can get a good representation for this lecture. Some of the characteristics of the disease cholera. Now what's interesting, they come around seasonally. So you have these seasonal epidemics, typically during monsoon season, where sewage from villages can wash into drinking water sources. That's just one way that happens. What's interesting, too, is that all ages are equally susceptible, especially if the individuals in the population lack prior exposure to the particular strain that's circulating around. And another important point is that the infectious dose is really high for cholera, and that's what I was alluding to before, that paradox of how these microbes can actually infect the intestinal tract when they're so pH sensitive. The answer to that is that the organisms have to be in super high density. So if you get, an amount of cells equal to 10 to the 11th, most of them are going to die through the stomach acidity, but enough of them will survive to get to the small intestine. An interesting experiment that can be done if you treat a bunch of, not treat, but if you expose a bunch of people to sodium bicarbonate, which is, basically like a antacid, that raises the stomach pH. In turn, that allows the infectious dose to drop dramatically, just showing you how absolutely important it is to have a low pH stomach. So again, raising the pH of the stomach with an antacid will actually lower the infectious dose. Fewer cells are required to infect the individual. The incubation period, as you can imagine, is usually about a day or so, but I've seen it much quicker than that, within 7 or so hours of initial consumption of contaminated water. Acute cholera cases may excrete 10 to the 7th to 10 to the 8th cells per gram of feces, which is, correlates to about 10 to the 11th to 10 to the 13th highly infectious cells, which sort of gives you the understanding of how these numbers can be attained in the environment, because these are astronomical numbers. And so if each acute cholera case is excreting this much material constantly, and it's not like it just, comes and goes, it's pretty constant. The location, the environment can rapidly build up, especially when it's torn through a whole village and everybody is excreting cholera at high levels and it's seeping into the water supply and so forth. The big reason why individuals die of cholera is this right here, that you can lose up to a liter of fluid per hour, causing severe dehydration and shock. Typically within 6 hours, an individual will die, and so if untreated. And so here you see an image of a hanging latrine in Haiti, and answering the question, how does this even happen? Like, how is there such a breakdown in public health measures that cholera can reach such levels in the environment? But here's what should be a beautiful tropical location, littered in garbage, and here's the one of the primary causes, this concept of a hanging latrine. You find these in Nepal and India and lots of places that have high densities of very poor people that don't have internal plumbing and all those things, and so they come to these public restrooms, which are nothing more than just shacks where they can poop down into the water below. And so here you see guys that are out here fishing. So if one cholera excreting individual can easily contaminate much of this area here that I'm highlighting in with my pointer. So then imagine dozens of people with the same affliction excreting tens of millions of cells of cholera per gram of feces. And it's hard to even call it a gram, because the excrement is called the rice water stool, and this is what would generally be excreted. It mostly looks like, as the name suggests, the water that's left over after you've cooked rice in it. And so, there's usually a little bit of solids in there. This is mostly just mucus and so forth in the intestinal lining. But then imagine this little baby that wouldn't be able to lose much fluid without actually dying. And so in this particular case, the little one is intubated and is receiving IV fluids to resuscitate. Getting back to the genetics, this is a representation of the toxar regulon of Vibrio cholerae. And so toxar is a transcription factor, and regulon is a term that is used in bacterial genetics to describe clusters of genes that are under regulation by the same regulator, or a common regulator. And so in this particular case, here's the outer membrane of the bacterium. Here's the periplasm, so in here would be where the cell wall of the bacterium is. And then this is the inner membrane, or the cytoplasmic membrane. And embedded in that is toxar, these dimers that you see here in red, and another protein called tox-S. And together, toxar and tox-S recognize environmental signals that activates them to become an active transcription factor. The signals that they're looking for include temperature changes or osmolarity, or things that are changing in the medium composition, such as bile salts, pH, and oxygen tension as well. And so all of this stuff is what happens when the microbe makes the transition from, for example, an estuary to an intestinal tract. There's changes in all of these things. And so the microbe has to be responsive to that. And basically that causes them to activate gene expression of the cholera toxin gene. Over here is the toxar and tox-S gene on a bit of the chromosome. And again, this is just, this would be contiguous pieces of DNA in those big circles that I showed you previously. But I'm only showing you sections here just so you can kind of get a sense for the gene expression cascade. So this is the promoter. Here's toxar and tox-S. So these genes are expressed, and ultimately translated into proteins that fit into the membrane here. Over here you see the cholera toxin genes, CTXA and CTXB. And so again, this is an AB type toxin with B domains for binding to the host cell and A domains that give the toxin its activity. When ToxR and ToxS recognize the appropriate environmental signals, they'll change conformation, and that will cause ToxR here to dimerize. So these little dumbbells will cross over each other, instead of being two parallel lines, they'll cross. That's probably not exactly what happens, but just imagine that dimerization looking something like that. In that dimerized form, they can activate, that's why I have a plus here, the promoter, P here, for the cholera toxin genes. They can also activate, over here, the promoter for TOX-T, which I've already talked about. And I mentioned that in that slide with the genomic circles, the genome representation. And I talked about VPI, and I talked about CTX phage. So by activating TOX-T, TOX-T can actually be produced and go over and equally activate, or probably more strongly activate, the promoter for cholera toxin. TOX-T also activates the promoter for toxin co-regulated pilus production. Remember that TCP is essential for the virus to attach and infect. And so again, TOX-T makes the cell more permissive to virus infection. Don't worry about some of these extra things over here. There's another transmembrane sensor protein complex, TCP-PH, that also can recognize these environmental signals and activate TOX-T. So there's multiple inputs in this circuit for ultimately getting elevated expression of CTXA and CTXB. So this is just showing you the mechanism of action of cholera toxin. So again, cholera toxin is an AB toxin. This is the normal process over here. And so in this particular case, this would be happening in your intestinal lining, in the cells of your intestinal lining. And it starts with ATP. And in this process, a series of enzymes, adenylyl cyclase, G protein, and an energy source come together to convert ATP into cyclic AMP. And cyclic AMP is the second messenger. It has a function in the cell. In this particular case, it regulates ion channels. So this comes together and converts ATP to cyclic AMP, and then the GTP is hydrolyzed and released. That's what you see here. And so here's the GDP and the phosphate, and then the G protein that goes from S to active form to I inactive form. Now you have adenylyl cyclase and the inactive form of the G protein. These guys are ready then to bind a new GTP and then grab onto a new ATP and make another molecule of cyclic AMP. So it's a very stepwise and controlled process, as it should be, because you don't want too much cyclic AMP, because you don't want the ion channels to just be completely open all the time. With cholera toxin, it's a little bit trickier. So over here is the adenylyl cyclase and G protein. In the presence of cholera toxin, there's the A portion of cholera toxin. These guys here, in the presence of the A portion of cholera toxin, grab an ADP ribose group off of NAD and sticks it onto the G protein. So that A portion of the toxin grabs ADP ribose from NAD and sticks it onto the G protein here. So then adenylyl cyclase, G with ADP ribose on it, grab onto the GTP here, and now you have this complex. Adenylyl cyclase, active G protein, the energy source, but this time with an ADP ribose group stuck to it. And in this conformation, the G protein can't be inactivated, as you see here. And as a result, this complex stays together and continuously just keeps making cyclic AMP. Oops. Jeez. continuously making cyclic AMP. And so this is an easier way to look at it. Well, here's cholera. I already showed you this picture. Here are the bacteria sitting on the microvilli of a small intestinal epithelial lining. And then imagine my pointer then is a toxin molecule. And so the toxin molecule would go over here and bang into one of these microvilli. And so there's the toxin molecule here. And here are the B subunits in blue and the A subunit in red. So this is the CT or cholera toxin. The A portion is ultimately what has the enzymatic activity, so it has to get into the cell. So as you see, the whole thing comes in an endosome, and then eventually the A portion is processed. And so that's what you're seeing here. There's a little bit of a cutting going on here. And so the A portion is processed and into this A1 fragment here. And then the A1 fragment can add the ADP ribose group to the G protein. And then the G protein, adenylate cyclase, and can overactively stimulate the production of cyclic AMP from ATP, as you see here. And so this cyclic AMP then will activate this protein kinase A, or PKA. And it's actually protein kinase A that phosphorylates these transmembrane ion channels. And so by phosphorylating them, it causes the CFTR, for example, to stay open so that it just leaks chloride into the lumen of your intestines. And by phosphorylating this channel here, it blocks sodium uptake. Same with this one, blocking sodium uptake. And so sodium ions and chloride ions and other stuff too build up in the lumen of your intestinal tract. And so hopefully you get the gist of where this is heading. By osmosis, water will always flow from an area of low concentration of dissolved solutes to an area of high concentration. And so the concentration of dissolved solutes here will be much lower than this salty, etc. environment here. And therefore water from this cell will flow outward massively. And when this cell gets dehydrated, then water from below will seep in and flow out as well. And so that's where the massive diarrhea comes from.